Hi, we will continue to explore the temperature-dependent sex determination in reptiles and particularly in turtles. And now we will begin more precisely to work on field when you have field uh, nest, how you can estimate sex ratio. So I said that we will work more specifically for turtles because we will place our discussion in the context of male-female pattern of TSD with male produced low temperature and females produced at higher temperature. So this is the most common pattern of TSD in turtles. We have also male-female pattern in TSD, for example, Chelydra serpentina, but it seems to be more rare. We have seen in previous videos that we can model this pattern of thermal reaction norm of sex ratio using different kinds of equation. And we concluded previously that the flexed double star model with Bayesian fitting was the best strategy. RNU effect can be included as fixed effect or random effect, but we concluded that fixed effect was the best solution and it can be tested using WAC. Other models can be implemented. For example, we saw the stairs model, but at this time we don't have a possibility to choose between flexed double star or stairs model. We have seen also in previous video that there is a special time during development where the sex determination is sensitive to temperature. This is a thermosensitive period of development for sex determination. The abbreviation is usually TSP, and it goes from stage 16 to 22 with Intema table, and from stage 23 to stage 27 with Miller table. Here I show the relationship between days of development and SCL straight carapace length and the correspondence in stages. This is data. These are data from Casca thesis, and you should take into account that this X comes from field nest, and then the scale of days of incubation are not homogeneous among nests. So this data can be problematic. That's why generally it's better to use Emis orbicularis table and a conversion between Emis orbicularis to marine turtle table. Then we have established when in Miller stages the TSP occur during development. To use a time series of temperature, we need to convert the TSP in stages as TSP in days of incubation. Then we need a model of a thermal reaction norm of growth of embryo, taking into account temperature. We show that we can use our Gompertz model and using as an embryometric straight carapace length, or progression of development using a linear model, and we concluded that the Gompertz model was better to fit our need. So here I show an example of model of growth of embryo with a temperature in green, with uncertainty of the growth of embryo shown as dashed lines. And you see that the TSP is completely shifted to the right as compared to the second third of incubation. So it is clear that the second third of incubation is not the TSP when temperature varies. It is a TSP for constant incubation temperature, but it is never the TSP from filled nest. This is one very common mistake in publication. So we have now different methods that are available. We know how to model the growth of embryo. We know to determine where is the TSP. We know what is the thermal reaction norm of sex ratio. And now we must use all this data to estimate the sex ratio from field for a natural nest. And the question is, what temperature obtained from field nest should be used to estimate sex ratio using the thermal reaction norm of sex ratio as a guide? The thermal reaction norm of sex ratio, which is the guide, is obtained at constant temperature. Then this temperature from field that we will estimate will be called a constant equivalent temperature. First, let's see what's happened when we take into account that embryo is growing at a different rate for different temperatures. During incubation, imagine that you transfer an egg in a freezer, and imagine that it is not lethal. You must have imagination because, of course, it is lethal. Such a low temperature is obviously masculinizing. But no development occurs at all in freezer, and then the embryo does not feel this low temperature. It will have no influence for sex determination. So what is important is the fraction of development that occur at different temperatures. This is not the average temperature. This is a measure that takes into account how much development occur at different temperatures. This has been proposed initially by Claude Piot in 1982 and perfectly demonstrated by seminal work of Arthur George and colleagues in two papers, 1999 and 1994. These papers are very important in our context. The experimental design was very, very nice. They incubated eggs at the same average temperature, but they varying amplitude of temperature. You see, you have here 4 different patterns, and all of them have the same 26 average temperature, but you have variation in amplitude of temperature. So the average temperature is exactly the same in all these experiments. If we report the observed sex ratio, here male frequency, according to the amplitude of incubation temperature in degrees Celsius, you see that the more amplitude temperature are high, the more feminized are the eggs. Then there is clearly an effect of amplitude of temperature and then an effect of growth of embryo. We must take into account this, and this experiment clearly invalidates the average temperature as a proxy of CTE. We must take into account also that sex determination occurs during a window of development. This window is the thermosensitive period of development. It proves that depending on the stages, the impact of sexualization of temperature will not be the same. Then we must include an effect of stage within the TSP on the capacity of temperature to sexualize. Sexualize, it means if you drive an indifferentiate gonad toward an ovary or a testis way, to sexualize the embryo. We call this effect the sensitivity of sexualization during the thermosensitive period of development for sex determination. Similar effect can be also put on beginning and end of TSP. You have also uncertainty on the beginning and end of TSP. The third point that we must take into account when we estimate CTE is that the Celsius measurement follows a relative, not an absolute scale. It means that the capacity of a temperature to sexualize is not simply a proportion of the quantity of heat. The thermosensitive reaction norm for sexualization parameter permit to take into account this property. For example, if you are at 30 degrees Celsius, you are at feminizing temperature. But if you are at 35 degrees Celsius, this is also a feminizing temperature, but the feminization is more strong. So the capacity of a temperature to sexualize the embryo is different, even if both temperatures are purely feminizing temperatures. So, in conclusion for this part, the TSD pattern at constant temperature is relatively well understood and modelized, but it is not the same for sex ratio on the field. We should take into account that TSP must be determined using thermal reaction norm of embryo growth. We must model CTE to take into account that sexualization of the gonad depends on many different parameters. It depends on the embryological stages. It depends also on the exact temperature experienced by embryo. Then it will be possible to estimate constant equivalent temperature for any time series of temperature within a nest. And this is what we will see in the next video. Thanks.